Hey guys, and welcome to chapter 15, which is all about the evolution of microbial life. And we're going to be talking a little bit more about like macroevolution. So we're going to talk about kind of like the major episodes of the history of life and how life actually originated. So Earth was formed about 4.6 billion years ago. And when it was formed, it was like this hot mass. If you can see, this picture kind of gives a rough idea of what it looked like. It was constantly bombarded by rocks and ice caused by intense heat and vaporized water. And so basically, because it was in such a harsh condition, right, and there was actually like no oxygen whatsoever, and really harsh chemicals and elements going around, because of that harsh condition, there wasn't any kind of living life. There wasn't any living life. So we had things like nitrogen and oxides, carbon dioxide, methane, ammonia, hydrogen, which don't sound too bad, but by themselves, that alone is not enough for life to happen. So eventually, after a while, the Earth started to cool down, and once it started to cool down, we started forming oceans. So this is again something taken by internet, but it went from something like having Earth that was full of volcanoes and bombardment and just lava, etc., into basically more oceans forming. And so once we had those oceans, which is basically 1,100 million years ago, life began. And the first sign of living life was actually a prokaryote. So that is, remember, the prokaryotes that we have today are like bacteria and archaea. And the prokaryotes that we had at the origin of life were even more rudimentary than the bacteria that we have today. So they evolved about 3.5 billion years ago when the Earth was still devoid of oxygen, so there's still no oxygen. And then a few of them started actually being able to make oxygen. So specifically cyanobacteria came into play, and they started to be able to make oxygen. And this oxygen that they now released made the atmosphere of the Earth, it basically added oxygen to it. So now we had oxygen in our atmosphere. And this oxygen production happened about 2.7 billion years ago as a result of these bacteria that were going through photosynthesis, because photosynthesis is the process that we very well know releases oxygen back into the atmosphere. They lived alone for about 1.7 billion years, and then afterwards they started having great abundance of different kinds of organisms. And so 900 million years later, right, after the prokaryotes, we finally got some eukaryotes. And remember, eukaryotes are cells that have, or organisms that are made out of cells that have nuclei and other kinds of organelles. So they first evolved from a prokaryotic community, and basically they, like things like the mitochondria, for example, I kind of already mentioned this to you guys in prior recordings, but the mitochondria, they assume came about because one big cell ate up one small cell. And then that one little small cell stayed and basically had an endosymbiotic hypothesis, or endosymbiotic, relationship with the bigger cell. And so that small cell became an organelle, specifically the mitochondria, which, as you guys know, makes energy for us. So this is also seen in chloroplasts. So this is a hypothesis that we use to explain chloroplasts as well. So same kind of scenario, but now the bigger cell that ate up a small cell or a smaller organism, the small organism ended up being able to photosynthesize, and so it became a chloroplast. So these multicellular eukaryotes evolved at least 1.2 billion years ago, and so we started seeing more complex things. Then even though they were more complex and had organelles, they were still kind of rudimentary compared to the organelles like the eukaryotes that we have today. And so we had kind of still unicellular, so single-celled protists. Those were basically the first eukaryotes that came about. So nothing like animals or anything, or plants or anything like that. The next great event in life was multicellularity. So once we had eukaryotes, they were single-celled, and then afterwards the eukaryotes, some of them became multicellular. This happened about 1.2 billion years ago. Even though they were multicellular, they were still quite small organisms and not very complex. And then after about 600 million years later, we had this huge event called the Cambrian explosion. And what the Cambrian explosion did is basically there was like an explosion of a number of different kinds of organisms that we had. So huge, like completely diverse and big, complex organisms showed up. And before then, they never were present. Okay. So after the Cambrian explosion, which resulted in the evolution of all major animal body plans and all the major groups, afterwards we started getting things like plants, fungi, insects, and they began to colonize the land. And then afterwards, after 65 million years ago, they adapted even more, and we got flowering plants, birds, mammals, primates as well. And then finally, about 195,000 years ago, we got our first modern human, the Homo sapiens. So I kind of went over that. Okay, but so let's look at everything kind of in a little bit more detail. So we talked about the origin of the Earth, like at the early Earth being just kind of this primordial soup. Like even prior to that, it was just actually like non-hospitable to life. And then afterwards, life started coming about because of the water vapor cooled and formed oceans. And we got these prokaryotes, right? These rudimentary prokaryotes. But in order for them to come about, we have to look at a few things. So first off, life is an emergent property that arises from the specific arrangement and interactions of its molecular parts. In this way, life is an example of interaction within systems. So in order for life to come about, you have to interact, like molecules have to interact together and stay together. And then the organelles have to interact together and stay together, that kind of stuff. That's why we went over the hierarchy of this particular, like in, I think it was like chapter 1 or something, of how biology works, right? So to learn how life originated from non-living substances, biologists draw on research from the field of chemistry, geology, and physics. So there's a 4-stage hypothesis for the origin of life. So basically how the first rudimentary prokaryotes kind of came about. So according to the 4-stage hypothesis, we have the first thing that happened was the synthesis of small organic molecules. And remember, organic molecules include things like, including like carbohydrates, proteins, DNA, that kind of stuff. So we had the synthesis of small organic molecules, so probably things like the subunits of those corresponding macromolecules, so like amino acids or nucleotides or something like that. Then these molecules had to actually come together. So the second step is the joining of these molecules to make bigger functional macromolecules like proteins and nucleic acids. Afterwards, these molecules have to be packaged into pre-cells. And then once we have a pre-cell, then we have this whole self-replicating process. A cell, in order for it to continue living, it has to actually replicate itself. And so once it was able to self-replicate, then that allowed for means of inheritance, means of passing on traits to offspring, and means of making more offspring as well. So the synthesis of the organic compounds, there was like a, they made an experiment, Stanley Miller in 1953, where he basically took all of what he supposed were the gases and the elements that were found on early Earth, and he mixed them together. And in this weird little contraption, he was able to make things like amino acids out of this weird little like sea of elements. The Miller experiment isn't actually, and I know your book talks about it a lot, but it's actually kind of incorrect, because the elements that he thinks were present in the early Earth actually weren't. We found out, like we now know that they're not actually there. So, but this still kind of gives an idea of these like synthesis of organic compounds from just random elements, from inorganic compounds. So we're constantly testing other hypotheses for the origin of these organic molecules, and it's just something that is still kind of out there. But once they are made, right, once the small little organic molecules are made, then the organic molecules have to come together to form that necklace, right, the polymer. And so they join together. This linking up is kind of random, so they had to like randomly come in contact with each other and join up. So it may take like millions and billions of years. So it is still kind of hard to wrap your head around, because it's basically like finding a needle in actually it's worse than finding a needle in a haystack, basically. But somehow the polymerization of monomers came about so that they formed polymers. So huge macromolecules like proteins and nucleic acids were made. They say that it was a combination of including things like hot sand, clay, or rock that aided, kind of allowed that warm environment, that hot environment to allow for polymerization to occur. Once that happened, these macromolecules all have to be found within one single cell, right? They have to be kind of enclosed and kept together. And this is where the formation of pre-cells happened, where basically these lipid-like molecules kind of formed around the organic molecules and packaged them up, and those were the pre-cells. And then once we had the pre-cells, we had to somehow have a way of replicating these cells, right? Because we had to replicate the macromolecules and the information found within the cells to make more cells and to have more offspring and to allow for the continuity of life. So how do scientists suppose this happened? The biggest hypothesis that they're going on right now is called the RNA world hypothesis, where they say that RNA could do anything. So RNA could not only hold the information, the inheritance information, which it kind of does since it reads the DNA, right? Right now we know that it reads DNA and it holds that information, but it also works enzymatically. So there's like we know that enzymes are basically predominantly proteins. However, some enzymes are called ribozymes are actually made out of RNA. So RNA can hold information and it can work as an enzyme. So because of that, they're thinking that the RNA was something that was necessary to allow for self-replication. So the RNA kind of duplicated itself and then it split up the cell into two so that there was a continuity of life. So from chemical evolution to Darwinian evolution. At this point, natural selection would have begun to shape. So once we have the first life, right, it's first sign of life, natural selection would have begun to shape the properties of the pre-cells that contain self-replicating DNA. The pre-cells that contain genetic information that help them reproduce more efficiently than others would of course have increase in numbers, right? If they have an advantage over other organisms, they obviously have an advantage over the ones that kind of lack those abilities. And then at some point during millions of years of selection, DNA was actually selected was selected as being better suited for the information flow molecule than RNA. And so the cells that had the DNA had that advantage. Okay, so yeah. So that's basically it. That's basically the introduction to life on Earth, right? And so now that we know how everything has kind of evolved, let's actually focus on the prokaryotes. Prokaryotes of what they used to be. Like, remember, they were the first organism to ever evolve, and also we're going to look at what they look like today. So prokaryotes lived and evolved all alone on Earth for about 2 billion years. To this day, they continue to adapt and flourish, and there's billions and billions of prokaryotes in this world. They've helped modify the planet, so they were the ones that actually, like a specific kind of prokaryote, allowed for the entrance and production of oxygen in the atmosphere of the Earth. And they're found pretty much everywhere. They're found in soil, they're found on your skin, they're found within your own body. They're found in the depths of volcanoes, they're found like in the atmosphere, they're found absolutely ubiquitously. So so they have a collective biomass of at least 10 times that of all eukaryotes. So there's a lot of prokaryotes, which include bacteria and archaea. And some of these prokaryotes are also really kind of like extremophiles, meaning they live in very extreme environments. So they can live in incredibly cold environments, incredibly hot environments, very salty environments, or acidic or alkaline environments that normally most eukaryotes would not be able to, actually the majority of eukaryotes would not be able to live in. So an example would be the Archaea halobacterium. This particular archaea can live in the Laguna Salada in Spain. This particular lake is saturated with salt. So you would go in there and you pretty much shrivel up like a raisin if you could. No human being would be able to live in such an environment. No normal eukaryote would be able to live in such an environment. But prokaryotes, since they have certain kinds of prokaryotes, since they have very unique adaptations, they can actually grow in incredibly non-friendly environments or in hospital environments. So due to their ability to adapt to diverse habitats, they're the most abundant organism found on Earth. Like I said, they're found ubiquitously. And some of them are completely beneficial to us. We like them, and others can actually be very bad for us, and they can cause all sorts of human diseases. So you may have even heard of a couple of infections that are caused by different bacteria. So you may have heard of Staphylococcus aureus, right? You may have heard of tetanus. You may have heard of any kind of pneumonia, those kind of things, meningitis. Those are all caused by various bacteria. Okay, so even though there's some really nasty, gnarly bacteria out there, not all of them are bad. And in fact, not all of them are bad, not just for the environment that they're in, but there's sometimes you need them actually in your own body. You may have heard that, have you ever heard of the words probiotics and how we need to take in probiotics? Probiotics are essentially bacteria because we want to replenish the stores of bacteria that we have in our own body. Our body has areas that have bacteria in it. Our mouth has bacteria, our gut has bacteria, our skin has bacteria. So we have various different tracts and areas in our body that have bacteria. And it's important that we maintain those bacteria there. because the bacteria have a really important role. For example, a lot of them in our GI tract actually supply essential vitamins to us. They can break down certain things for us, right? They allow for the extraction of nutrients from foods that we could otherwise not digest. Yeah, the ones on our skin, they decompose dead skin cells, and they especially guard against disease-causing intruders. So I kind of like to a picture of bacteria, like especially the easiest example to give is the one on your skin. So you have a film of bacteria covering your skin. And I know that sounds disgusting, but remember, this bacteria is good bacteria. And so since you have a film of bacteria that's covering your skin, no other bacteria from around your environment can actually situate itself there, because otherwise it would be in direct competition for shelter and food with the bacteria on your skin. The same kind of concept or principle can be directed to our gut microbiome, our gut bacteria. And so it's really important to have them. They play a very crucial role in our defense, in defending us against possible pathogens. In addition, they also, like our gut microbiome, there's been a lot of studies that say that they are directly related to obesity, they're directly related to neurological functions and mental disorders. So it's really important to keep a healthy gut microbiome. And when I keep saying microbiome, that just means community of microbes or community of bacteria. Okay. Outside of our bodies, prokaryotes are really good because some of them, like especially in the soil, they help decompose dead organisms. Otherwise, we'd have like carcasses of like plants and organisms and everything kind of just, and other organisms just pile up on the world, and our world would just be like just full of trash. So thankfully, we have a lot of decomposers that are prokaryotes, though not all of them are prokaryotes, that can decompose and kind of recycle this wasted material and recycle the material back into the earth as good elements. So they have a very important role. So what do prokaryotes actually look like? So they are very different looking than eukaryotes. First off, remember, they don't have any kind of organelle, so they don't have any compartments within themselves. So they don't have a nucleus, they don't have mitochondria or anything like that. So yeah, they typically have a cell wall, which is just an external extra wall around their plasma membrane. And even though they look quite simple, they're actually a remarkable range of diversity. Like I said, there's billions of prokaryotes, and each one is different from the other. So they are, even though they're single celled, they don't have any compartments or basically organelles, they are still quite, like within their metabolic actions, their metabolic chemical reactions are super different from one another. Okay, but generally, when you compare a prokaryote to a eukaryote, prokaryotes are very small compared to eukaryotes. Prokaryotes lack any kind of nucleus or organelle compared to eukaryotes. Prokaryotes do have ribosomes, because remember, ribosomes are the sites of protein synthesis, so they need to make proteins by them as well. And so if we got rid of, if prokaryotes didn't have ribosomes, prokaryotes wouldn't live. So prokaryotes and eukaryotes have ribosomes. They tend to have a cell wall around them, the prokaryotes, and they just, they have typically one chromosome, one circular chromosome as compared to eukaryotes, which can have multiple chromosomes that are usually linear. So we characterize these prokaryotes in different forms. The forms usually, so we put them in different kind of categories. The categories are usually dependent on their shape. So the shape of the bacteria when analyzed under a microscope. So the spherical ones we call cocci, the rod-shaped ones we call bacilli. I like to think of, like in Harry Potter, you know, the snake, the basilisk. The snake is kind of a rod-shaped shape, and so the bacilli kind of sounds like basilisk. The bacilli are a rod-shaped bacteria, and then we have the spiral bacteria, like this one right here, that are spirochetes. So different kinds of bacteria are spherical or rod-shaped or spiral. For example, Staphylococcus aureus. The word coccus in it is because it is spherical. E. coli is, or like you probably have heard of Escherichia coli, that is a rod-shaped bacteria. And bacteria that is spiral is something like Treponema pallidum, which actually causes syphilis, or even Borrelia burgdorferi, which is the one that causes Lyme disease. So there's plenty of different, each bacteria falls into one category, and we like to categorize them based on their shape. Or one of the ways we like to categorize prokaryotes is based on their shape. So even though they're unicellular, they can, they are diverse, and you can categorize them in different types of things. sometimes they actually like to stay in more than one, they prefer to stay in the masses. Bacteria have really realized that in order for them, like a clear advantage of their survival in the environment is that they stay with the masses, as opposed to staying alone. So a lot of times you see most of these prokaryotes kind of stuck together. So this is why we see many different spherical bacteria here. There's many of them. Each of these is just one single individual bacterium, or like many rod-like bacteria kind of sticking together, because there's usually strength in the masses. Another thing is about half of the prokaryotic species are mobile, meaning they can actually move. And the major way that they can move is with the use of a flagellum, which is just a tail, right? It's with the use of a flagellum. These are other kinds of, this is just kind of bacteria that underlines the diversity of bacteria. We have things like actinomycetes, cyanobacteria, which can photosynthesize. And as you can see, they stay together like so. Each one is individual, but they can stay together like so in a chain. We even have bacteria that kind of go against the normal rules of prokaryotes. So an example is a giant bacteria like Epulopaecium thistlestoneae. Epulopaecium thistlestoneae can actually be visible to the naked eye. So it's actually pretty much the size of a semicolon in your textbook, which is very crazy compared to other bacteria, because most other bacteria you cannot see unless you visualize them under a very high magnification lens in your microscope. So that just kind of shows you the great diversity of prokaryotes. Like there's not all of them fit all the rules, and it just again contributes to the genetic variation and diversity of the prokaryotes. A lot of times, remember how I said there's strength in the masses? So most bacteria or prokaryotes like to attach themselves to a surface and kind of form this community of bacteria. And this is known as a biofilm. So they kind of colonize millions of bacteria, go ahead and colonize the surface, and they form a biofilm, which can consist of millions and millions of different species of prokaryotes. So a biofilm is a surface coating colony or one or more species of prokaryotes that engage in metabolic cooperation. So if you've ever been in the shower and you feel like what you think is soapy scum on your shower, on the bottom of your shower, that's actually a biofilm of bacteria. Any kind of dental caries or cavities, that's actually a biofilm. That plaque is a biofilm of bacteria found within your tooth. So they decide to stick to your tooth, and they accumulate other bacteria to kind of form that to make that community bigger. And in fact, they actually even go ahead and harm your teeth in that way, but that's a different story. This is just examples of how bacteria like to form biofilms, and that they're kind of primed to do it because, again, there's strength in the masses. So they just like to recruit nearby cells to basically grow the colony. In addition, the cells that are found within the colony go ahead and specifically produce sticky proteins or sticky sugars like polysaccharides to allow for everything to just be a little bit more sticky, so that more cells can just stick and just stay stuck on the biofilm. Within the biofilm, it's a community. So you can actually find channels and tunnels across the entire biofilm. So you can see here, channels across the entire biofilm so that bacteria can communicate with each other even though they're far away, and they can send specific metabolites and nutrients and minerals down these channels. Biofilms can occur pretty much anywhere on any type of surface, like rocks, organic material like living tissues. In fact, 2/3 of any kind of bacterial infection in your body is actually from a biofilm. They can do it on metal and plastic. That's why it's very important to make sure that in a hospital setting, for example, everything is super clean, because we don't want a biofilm and bacteria to form on it. Okay. The way prokaryotes reproduce is actually very simple. It's known as binary fission, and binary fission is just a method of asexual reproduction. So they don't need a counter partner in order to sexually to reproduce themselves or to make more of their offspring. They can just reproduce asexually, in which case a single-celled organism grows to roughly double its size and then divides into 2 cells. So essentially what you're doing is this, like the cell grows, and then once it's big enough, it's just going to pop into 2. So before this happens, of course, your DNA has to replicate. So if this is my DNA, it replicates into 2. I know they're 2 different colors, but we're just going to deal with it for the moment. The DNA gets brought to the opposite sides of the cell, very simple, and then the cell, like this growing cell, just splits into 2. It's incredibly simple, much more less complicated than what we see with eukaryotes. Okay, and so because of this binary fission, because the way they reproduce is so fast, they can grow their populations really quickly as well. In fact, E. coli replicates itself every 20 minutes. So every 20 minutes, E. coli makes two daughter E. coli, two progeny. So that's crazy. The reason why they can reproduce and make such rapid growth in populations is because A, they're very small. Two, they use a very easy way of asexual reproduction called binary fission. And B is because C is because they have short generation time. So they don't, unlike humans, they don't have to wait 9 months. Honestly, most of them wait about 20 minutes to half an hour to reproduce. So they're very quick to reproduce. And so therefore, a population of prokaryotes consists of trillions and trillions of individuals. So yeah, trillions and trillions of individuals. However, keep in mind that some of this population, after like this exponential growth, can become stationary. Like it can be, the ones that will grow, that as more bacteria are made, some bacteria is going to die. And so basically the population kind of levels off because everything kind of cancels each other out. In addition, some prokaryotes are just very good at surviving. Some of them can form things as endospores, which are basically like little army tanks. So they're just thick coated coverings that the bacteria does on itself. So this is a bacteria and it forms an endospore, and you can see how many coverings it has. And with these endospores, these thick coated resistant cells that some bacteria can form, they are able to withstand really insane harsh conditions. So any kind of disinfectant is not going to get rid of endospores. Normal, heating is not going to get rid of endospores. Antibiotics as well have a hard time getting through endospores. Even radiation, like endospores can even withstand radiation. So this is just certain kinds of bacteria are able to form endospores where they can kind of hide themselves in this little army tank, the endospore itself, and stay there until the environment around it gets better. Okay. So they're very, very durable. In order to actually kill them, we have to heat them to 121 degrees Celsius at very high pressures, which is basically the conditions of an autoclave. So not normally, with normal conditions, you can't kill them. And once the environment improves, then they rehydrate themselves, the endospore goes away, and the cell comes back out, and it basically resumes its normal metabolic activity. Prokaryotic nutrition. So like I said, prokaryotes are very diverse. And even though they don't look very diverse, they really are. And it's all because of the way they actually get their nutrition. They're a lot more complicated than humans or animals. All animals basically eat their nutrition, right? And eat something, food, in order to actually gain energy from it. Well, prokaryotes, it's not that difficult. It's a lot more difficult. They have pathways that transform energy and matter that are far more diverse than those of eukaryotes. Some species can get energy from things like ammonia and other things from like hydrogen sulfide, not from things just like sugar. Some can also get it from sugars, just like us, but sometimes they can even get it from really weird inorganic substances as well. Okay, so this extensive variation in prokaryotes is reflected in their diverse nutritional adaptations. So what are the categories of prokaryotic nutrition? So we have phototrophs, which obtain energy from light. We have chemotrophs, which obtain energy from chemicals. We have autotrophs, which basically require carbon dioxide or related compounds to make as a carbon source. Heterotrophs, they require organic stuff in order to get organic compounds like sugars to get their carbon from. Okay, so we have all sorts of things, and you can even put them together to give you the different types of nutritional modes. So we have photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. So I definitely suggest you look at this table. So because of their crazy, amazing metabolic talents, prokaryotes make excellent symbiotic partners with animals, plants, and fungi. That's why the fact that we have them living in our gut is so amazing. In fact, I don't know if you guys know this, but we have more bacterial cells than we have animal cells. I think if you weigh the amount of bacteria found in your body, it's about 2 kilograms of bacteria. That's a lot of bacteria. Okay. So because remember, so the symbiosis, well, we're just saying that because they have great, amazing talent in obtaining nutrition, they're really good at forming symbiotic relationships. And what symbiotic relationships are just relationships where both the organism, like one organism and the other organism, are benefiting. So they're just living together, and when they're living together, they're closely associated and they're both benefiting from it. So the host, right? So in our case, in the example of like the human and the bacteria relationship, we are the host and the bacteria is a symbiont, and we both benefit. Particularly the bacteria benefit because they're nice and safe in a human body, and there's not really going to be any bad pathogens coming at them, like any bad predators coming at them. And we benefit because the bacteria do also a myriad of different things, right? They help us digest different things. They help us make vitamins. They provide us with different vitamins. They protect us from some invaders. So this is basically what's known as a mutualistic symbiotic relationship where both organisms are going to benefit. There are other relationships like commensalism. Commensalism is when one organism benefits and the other one isn't even like, doesn't even realize that there's another organism living in it or near it. Parasitism, that's when one single organism benefits and the other one does not. The other one's actually harmed. So a parasite, it's going to, obviously if a parasite eats you or you, like it's living in you, it's going to harm you, and in itself, like the parasite itself is going to flourish. And so a lot of pathogens are considered parasites. Pathogens are just disease-causing microbes, essentially, usually. Okay, so because of the ability of just the nutrition, the different types of nutrition uptakes that they have, because of their ability to undergo these symbiotic relationships, prokaryotes are very important for the existence of some ecosystems. Without them, they wouldn't, like the ecosystem wouldn't come about. So for example, like we talked about cyanobacteria, if cyanobacteria were not photo or autophototrophs, they wouldn't have made oxygen. And so if we didn't have oxygen, oxygen would never have been introduced in this world, and no other living life would have, been made. No life that requires oxygen would have been made. So that just gives you an idea of how important some of these microbes really are. There's also an example here of the hydrothermal vents. That a lot of the hydrothermal vents are basically found in the depths of oceans. And basically they require bacteria for energy. So they're found in the depths of oceans. There's no sun, there's nothing down there. So bacteria that live there are the only source of energy because there's no sun. And so energy is derived from metabolic activities of bacteria that can actually take chemical energy from compounds like hydrogen sulfide that are basically found in the hydrothermal vent. Okay. So as a result of their nutritional diversity, prokaryotes perform a variety of ecological services that are essential to our well-being. Life depends on the recycling actions of chemical elements that like prokaryotes do. They play a very significant role in chemical cycles. And then they also break down, like I said, they can also be decomposers, and they can break down different dead organisms and organic waste. So very important for the environment. We can also use prokaryotes for our own benefit. So one really active study that one really active field of research that was happening right now is actually the field of bioremediation, where we're trying to remove things like pollutants from the water, air, or soil by actually using biological means. And that biological means are going to be your prokaryotes. So we're trying to look at prokaryotes that can decompose or treat sewage, that can decompose or treat different kind of other pollutants. So it's a really big deal because we want to get rid of, it's a way of us cleaning up these toxic chemicals, and not just to clean them up, but to also recycle good elements from the toxic chemicals. If they're used by, if a prokaryote starts using those toxic chemicals, it can then spit out good waste products, so good elements that we can use. So it's just, yeah, this is an example is actually a lot of times when we have more and more recently, if there's any kind of oil spill in the ocean, bacteria are released that specifically digest and eat oil for energy. So that black oil for energy. So it's a very big thing, and it's again ongoing research is needed because not a lot is known, and it's something that we definitely have to work towards because bioremediation is, the amount of pollution that we have coming into the environment is preposterous, right? Okay. Right. So we have other kinds of prokaryotes as well. So remember, we have 3 domains of life. The domains are bacteria, archaea, and eukarya. Bacteria are prokaryotes, archaea are also prokaryotes, and eukarya are eukaryotes. So we have 3 domains, bacteria, archaea, and eukarya. And then the domain eukarya kind of splits off into different kingdoms. And then we'll look at these kingdoms later on in like upcoming chapters where we have like the Protista kingdom, the plant kingdom, the animal kingdom, and the fungi kingdom. Okay, so I think now we're going to talk about archaea. So archaea are kind of new. We recently discovered them. They were first kind of discovered at the end of the 1970s. So not much is known about them. Another reason why not much is known about them is because most of the time, even though they're found ubiquitously, they're found pretty much everywhere. They're even found within your teeth, like inside the, like if you have like any kind of cavity, you can have archaea there. But most of the time, these archaeal organisms, which is just a different kind of prokaryote, live in extreme environments. So environments that are very, very hot or very, very salty. So yeah, they're abundant in many habitats, but most of the time, like they're especially known for being extremophiles, where they live in extreme environments. So they can be extreme thermophiles. To be a thermophile, philia means love, right? So thermo, they love temperature, they love high heat. Other ones can be halophiles, where they like salt, kind of like the ones that we saw in the Laguna Salada in Spain. So they really like salt. So these kind of archaea could live in places like the Dead Sea, right, which is so salty that most animals do not live in it, right? But archaea can. Or like in the Great Salt Lakes, right? There's going to be archaea that live in those areas. There's also a special group of archaea known as methanogens, and these are basically anaerobic, so oxygen-free organisms. So they live in places that do not have oxygen, and they release a lot of methane. And so what we actually usually find them in we find them a lot in the guts of different animals, and they release methane, and therefore the animal is going to release methane. And they're really important because this release of methane is actually going to go up in the atmosphere, and methane is actually one of the biggest contributors to the greenhouse effect and therefore to global warming. So it's a really big deal to actually study these archaea. Okay, so they're found in the digestive tract of animals, and they're also found at the bottom of muddy lakes at the bottom of the the bottom of the lake in the mud, because that's anaerobic. So though some of them do live in extreme environments, not all of them do. Some of them live in very moderate conditions. Like I said, some of them live in your teeth. A lot of them live in the soil. So they're found everywhere. They're just not found in a lot. They're not found in as abundantly as bacteria, but they are found everywhere. Okay, so that was a little bit on, archaea. Now moving on to just some more stuff about bacteria. So some bacteria can cause disease. Remember, the ones that can cause disease are known as pathogens, and the diseases there's all sorts of different kinds of diseases. You may have heard of whooping cough. You may have heard of anthrax. Again, meningitis, pneumonia. You may have heard of endocarditis, which is just basically the infection of the heart. So there's all sorts of, or tetanus, leprosy, tuberculosis, cholera, gonorrhea, all sorts of different things can happen because certain bacteria are really, really bad for us. So they can be really bad for us because of many of these bacteria actually secrete specific toxins, or they have within them some toxins. So exotoxins are the toxins that they get secreted out into the environment. Endotoxin is just when they're part of the actual bacterial cell structure itself. So for example, in Clostridium tetani, which is the bacteria that causes tetanus, actually releases the toxin tetanospasmin. And that's a toxin that then goes ahead and it specifically inhibits like nerve signals from exchanging signals in your body. And so essentially your body no longer knows how to stop these nerve signals. And so your body keeps on contracting, but it never relaxes. And so with tetanus, you have these full body spasms that can get really dangerous, especially when you are of a young age. Endotoxins, an example of that is there's all sorts of particular bacteria that have a specific endotoxin known as lipid A. Lipid A is part of the actual bacteria itself. So as the bacteria is causing mayhem, and we want to get rid of them, and we usually get rid of them with something like antibiotics. We usually get rid of bacteria with antibiotics. The problem with getting rid of the bacteria that have specific endotoxins is that if the antibiotics kills all of them in one go, the cells of the bacteria are all going to burst. And if they burst, the endotoxin that they are keeping is going to be released in the environment. And that will be actually very bad for us. So this is why a lot of times with antibiotics, this is one of the reasons at least, with antibiotics, you actually need to take them in small doses for a longer period of time so that we can kill the bacteria slowly, and not just in one go. Because if we kill it all in one go, all of the endotoxin is going to be available in the environment, and that could be a very dangerous thing. And that can lead to, basically, with these particular endotoxins like lipid A, it can lead to a dangerous drop in blood pressure, right? Vasodilation, and then eventually shock. So you can die because of shock. But yeah, all of these are just other examples of diseases that are caused by bacteria. We have Bacillus anthracis that causes anthrax. Oh, this is my not my favorite one, but it's a fun one. Yersinia pestis, that's the one that causes the plague. All sorts of bacteria can cause it. But remember, not every single one is bad. In fact, we were just talking about how most bacteria we actually need in the environment, because without them, the environment would basically would no longer work. They are one of the main decomposers that we have in our world. So not all bacteria are bad. However, the bad, harmful bacteria, we want to make sure we get rid of them. And so to get rid of them, we take antibiotics. So things like words like penicillin, doxycycline, tetracycline, sulfonamides, metronidazole, those hopefully are words maybe that you have heard, right? So these are just chemical compounds that actually go ahead, that are toxic to these bacteria, that kill the bacteria. The first antibiotic that was discovered was penicillin. Prior to that, people were dying from really simple bacterial infections. And so thankfully, with the advent of antibiotics, we were able to get rid of them and save a lot of lives. Okay. The problem, however, with antibiotics is that now they're being overused. And so because of this overuse and abuse of antibiotics, a lot of bacteria are actually developing resistance. So you may have heard of something like MRSA. MRSA is methicillin-resistant Staphylococcus aureus. And this is basically where Staphylococcus aureus gets resistant to the antibiotic methicillin. And so we have to use other antibiotics. And it's getting more and more like a problem. It's becoming more and more of a problem because we're starting to run out of antibiotics that we have made or discovered. And so we have to figure out different means. So because of that, there's a lot of stringent rules as to how and when one should use antibiotics. That's why in at least the Western world, to take antibiotics, the doctor needs to specifically transcribe the antibiotics to you. They'll just you have to make sure that you actually follow the correct regimen of the antibiotics. So if it's like for 2 weeks, you do it for the full 2 weeks. You don't go over, you don't go under. So it's really important to practice proper use of antibiotics in order to make sure that we avoid the surge of these resistant bacteria from coming up. Okay, so this is why they said a third defense against bacterial disease is actually education, because people need to educate themselves and realize why it's important to follow what the doctor actually says and take the antibiotics properly. A lot of people even sometimes they have a little bit of they have the common cold, which are usually like viral. Common colds are usually come about thanks to viruses, and they'll go to the doctors or to the pharmacy or whatever and insist for antibiotics, when antibiotics, like we just looked at, they only work against bacteria. They're not going to work against viruses. Antiviral drugs work against viruses. So they don't work against viruses, but people will take them, and because of this, they'll make the bacteria just kind of have a chance to develop resistance against the antibiotic. So it's really important. Another thing that's important is basically prevention of these diseases. And that's just, normally it includes like good personal hygiene, washing your hands, good sewage treatment in the actual cities or whatever. So just general good public health regulations go a long way in preventing a lot of diseases, like infections, like bacterial infections. Okay, so now we're going to move on to protists. Now protists are going to be, remember, when back, in the early Earth, prokaryotes were our first organisms that came about, and then slowly unicellular protists came. And protists are your eukaryotes. These are the first eukaryotes that showed up on Earth. These are very, the the fossil records indicates that eukaryotes evolved after prokaryotes around 2 billion years ago. These primal eukaryotes were the predecessors of the great variety of the modern protists and the modern eukaryotes like plants, fungi, and animals that we have today. So they are part of the eukarya domain. And so because of that, they are considered eukaryotes. So they have a nucleus, they have other organelles. And from these protists, all the other eukaryotes came about. So animals came from them, more protists came from them, plants came from them, and fungi came from them as well. There's no great way of actually like giving a satisfactory definition of a protist because it's not really they don't really have a lot of things in common. The only thing that protist organisms have in common with each other is that they like moist environments. That's pretty much it. So if we find a eukaryotic organism that is not an animal, that is not a plant, and that is not a fungus, we put them in the protist category. So if basically the definition of a protist is a eukaryotic organism that does not have all the defining characteristics of a plant, an animal, or a fungus. Why do we have such an unsatisfactory definition? This is because protist was used as a label of convenience rather than a label that reflects evolutionary reality. It's convenient to have a single name for everything that's different, and this convention is used in the textbook but protists are actually many different evolutionary lines of organisms. So there's all sorts of different protists. There's like choanoflagellates, algae, all sorts of different things. So if you look here, this is the bacteria domain, the archaea domain, which we've already looked at. And then when we look at the eukarya domain and we look at the protists are not, they're not just one little branch of their own. They're all sorts of different branches, but they're not plants, they're not fungi, and they're not animals. Okay. So they're very, very diverse. So we can have protists that are algae. If the protist is an algae, that means it can photosynthesize and it can produce oxygen. So anything like small algae, like what the krill eats, basically phytoplankton, those are algae, those are protists. Anything like even big algae like the sea kelp that we find in our oceans here, those are different types of protists. We can also have protists that are heterotrophs, so that means that they specifically need to eat other things. Some of these heterotrophs are bad, right? So there are some parasites, there are some parasitic pathogenic protists out there. You may have heard of malaria. Malaria is caused by Plasmodium falciparum, and Plasmodium falciparum is a protist. Or in this particular example, in this picture, this is actually Trypanosoma cruzi, or no, actually this is Trypanosoma brucei, sorry, Trypanosoma brucei, and that causes African sleeping sickness. So there are some parasites, and then of course there's others that are not parasitic at all, that are just fine, and they live in our ponds, and you know, they just do all sorts of things. An example would be something like Euglena. So some protists are also mixotrophs. Mixotrophs can go through photosynthesis and heterotrophy as well. So they can both make their food and ingest their food. And actually Euglena is one of those where it can, see how it's green? It's green because it has chlorophyll in it, so it can go through photosynthesis. And in addition, it can also eat other things. It eats food. Okay. So they live in all sorts of diverse habitats, but when we look at the habitats, you'll notice that they have one thing in common, that they're all basically related to water. So most protists are aquatic. They live in oceans, lakes, and ponds. If they are found in the soil has to be damp, right, and moist. Otherwise, they live within our bodies or within animal bodies. And again, in a body, it's damp, it's full of water, right? So they need water. We can basically put protists into 4 informal categories, even though the classification of protists is still kind of a work in progress, but we organize them as best as we can. These 4 informal categories are known as protozoans, which are basically the ones that eat other things, slime molds, unicellular and colonial algae, which are the ones that can photosynthesize, and then seaweeds. Okay, seaweeds are going to be your multicellular algae. So the first one that we're going to look at are the protozoans. They're going to thrive in all types of aquatic environments, including your own body, because remember, your body is made predominantly of water. Most species can actually eat bacteria or other protozoans. So protozoans eat and ingest food. They can also absorb food from like dissolved water. A lot of protozoans can actually live as parasites in animals. And then they can cause a lot of like harmful diseases. So here we have, like we already looked over the one that causes malaria, and we already looked over the one that causes African sleeping sickness. This is like Giardia, which can cause like Camper's diarrhea usually. It's actually prevalent here in the US as well. This one right here, I can't actually see the name. I think this is Trichomonas. So this causes trich. Amoebas are also a type of protozoan, and there's some really gnarly amoebas out there. You can have like Entamoeba histolytica, which can cause like normal things, like not normal things, but bad things like amoebic dysentery and like internal bleeding, sorry. Or we can even have amoebas that can literally eat away at our brain. So we have like flesh-eating, brain-eating amoebas as well. And a few people have actually died because of that. So protozoans are not fun at all, okay? A lot of the protozoans are flagellated, meaning they have these flagella, these tail-like things coming out of them so that they are they can move around and they're mobile and they can move to different things. Some of these protozoans are non-parasitic, and so they're free-living and they don't require like to be parasitic on a host or like or a human or an animal, but a lot of them can be. So yeah, some of them can even be beneficial. So for this example right here, there's different flagellates that reside in the termites, in termites' digestive tract, and it actually helps termites break down cellulose. Amoebas are also motile, even though they don't have a flagellum. They're actually motile because of what we call these things known as pseudopodia. And pseudopodia is the ability to basically stream their cytoplasm in different directions. I'm going to mute this and show you guys a video. And as you can see here, it's streaming, it's moving the gel-like cytoplasm in different directions to form these pseudopodia, which are just these extensions to move the amoeba or the organism in one specific area. And so these new things, as you see how it's always moving, these new things are known as pseudopods. And the word pseudopod actually means fake foot because they're forming these fake feet in order to travel. They can even use these pseudopods to kind of go around their prey and ingest their prey. And so it's just really fascinating to see. So not all protozoan are necessarily flagellated. Some of them can, they still are motile, but they have other means of motility like this pseudopod cytoplasmic streaming. Yeah. We also have other protozoans that have pseudopodia, but the pseudopodia come out of like a little shell. So these are known as forams. So forams have a porous shell around them, and as you can see, it's full of these little holes. And they can release, like they can kind of jut out these pseudopodia or these pseudopods out of the holes in the shell. Yeah, it's really interesting because the largest type, this is technically, these are known as xenophyophores, and xenophyophores are single-celled forams. They're single-celled, so there's only one nucleus in all of this, but it's a giant cell that can obviously be seen, and it's found in a marine environment. Okay, so the pseudopodia extend through the pores in the shell, and they're used for swimming, shell formation. I'm just going to say shell formation and feeding as well. Another kind of protozoan are the apicomplexans, and the apicomplexans are all bad. All of them are bad. So these are pretty much the majority of bad protozoans fall in the apicomplexan subgroup. So this includes Plasmodium, which causes malaria, Toxoplasma, which causes toxoplasmosis, which is something that probably a lot of pregnant ladies have heard of. So this is why pregnant ladies have to stay away from like cat litter and uncooked meat. And this is because they could accidentally get toxoplasma, which can eventually actually kill the unborn child because the parasite can be passed on from the woman to the unborn child. Okay. We also have ciliates. Ciliates are really great protozoans because these guys are all good. They don't harm at all. They're just really cute. Ciliates are called ciliates because they have cilia, so it's in the name. Cilia are just like these little hair projections that allow for for the organism to move. So they can provide movement for the protist, and they can even, they're just like little hairs like this move. They can even move food towards the protist's mouth. So they're non-parasitic. An example would be Paramecium. And so this is an example right here, Paramecium. You can see it's moving because of the way the cilia are like fluttering, right? They actually move in like rowboat, like they kind of work synchronously, and it's as if they're rowing a boat. They're like, they kind of work like the oars of a boat or of a canoe or whatever. And this is how they move it. And so these usually ciliates are both heterotrophs, and they can also be phototrophs as well. Slime mold, this is another kind of protist. These are multicellular protists related to amoebas, and they feed on dead plant material, so they're really great decomposers. There's two distinct types. There's plasmodial slime molds, and there's cellular slime molds. These look actually kind of disgusting. Like if I would come across them, I would walk the other way. They just look really disgusting. But so the plasmodial slime molds, they can form these feeding bodies, which is what you see here, and this is what they use to like eat stuff. They extend pseudopodia among the leaf litter, and they eat up the decaying material. They can form a web-like form. Like they can form like this really long web-like kind of stretchy material that kind of increases their surface area and their ability to stay in contact with the food. So they kind of just like go around their food and just like eat it up. I'm not going to show that because it's gnarly. Cellular slime molds are very similar, but they have a feeding stage that is actually like solitary, like where they're just unicellular, but then when food is in short supply, the cells come together, they swarm together, and they form a slug-like colony. So anything that you see, like a bigger thing that you can see with the naked eye, that's more than one cell, and that's when they come together because they realize that the environment is getting harsh, and so they need to come together to like kind of protect themselves. Then we have unicellular algae and colonial algae. So these are these are protists that can photosynthesize, and they are unicellular, so they're made out of one cell. They're usually found in freshwater and marine ecosystems. We're currently trying to harness their ability to convert light energy to chemical energy for another purpose, to produce biofuels as well. So these are very important. They basically make up all the phytoplankton, which, as you guys know, is like the base, like the fundamental food in the food web of like any kind of marine and any kind of marine food web. They include things like dinoflagellates, diatoms, green algae. Dinoflagellates have just kind of like around them, they're just reinforced by plates made out of cellulose. Diatoms, actually it's very interesting, diatoms have the same component known as silica, which is the component that we find in glass. So they look really interesting. They look like really fun geometrical shapes. These are diatoms. And then of course we have green algae, which are named for the fact that they are green, right? They have chloroplasts in them and chlorophylls, and so they can absorb like energy that way, sunlight energy that way. And so we have these dinoflagellates, green algae, and diatoms. Dinoflagellates are interesting because you may have heard of something like the red tide. They're actually involved in that. So when there's too many of them, they cover the entire surface of the waters, and they give a really distinct kind of coloration to it, like red or green or whatever. And I think you've actually had red tides happen in Maine sometimes. And these are really bad because once the dinoflagellates die and they get decomposed, the decomposers take up oxygen in order to actually kill and eat up the dinoflagellates. And this leaves places of like where there's no oxygen. So oxygen, oxygen dead, they're known as dead zones in the ocean. And so this is really bad for our fish because there's no oxygen in our oceans and the fish end up dying. So it's a really big deal. And usually they grow in they get more and more when there's some sort of like waste product coming from like a chemical factory or something being like the runoff being put into the ocean. So the dinoflagellates like grow. Okay, hold on. And these are just like more pictures like the dinoflagellates, the diatoms, and the various green algae. You can see them kind of in this video, just all interacting together and being amazing and amazing photosynthesizers. So they're the main, these guys are one of the main reasons why we have oxygen in addition to plants, but another way of contributing to the oxygen in our atmosphere is thanks to these guys. And then finally we have seaweeds, which are very easy to remember, right? We eat seaweed, some of us. And so seaweeds are large multicellular marine algae that grow on rocky shores. They're very similar to plants. So they have like convergent evolution where they look similar to each other, but they're not related to the plants. They're actually more close, like they're not as closely related to the plants as they are compared to unicellular algae. So they're more closely related to unicellular algae than they are to plants. There's all sorts of seaweeds. We have green algae, red algae, and brown algae, which is basically your sea kelp. And so they look something like this, right? Which should be kind of familiar to us as well. So that's it for chapter 15, and I shall see you guys in the next video. Bye, guys.